My name is Daniel Cook, and I'm an anesthesiologist scientist at Weill Cornell Medicine, funded by the FAER Mentored Research Training Grant. I aim to better understand how the brain works by studying communication between cells in the brain, known as neurons. Communication by neurons is necessary for everything our brain controls, like sensations, movements, and thoughts. Neuronal communication depends on calcium coming into the cell, which triggers the release of chemical signals at connections between neurons, called synapses. So, understanding how synapses are controlled is crucial to learning how the brain works. The concentration of calcium in human beings is usually kept in a very narrow range. However, how this normal set point of calcium level outside the cell affects the control of communication between neurons is not well understood. I discovered a surprising, powerful effect on neuronal communication caused by relatively small changes of calcium concentrations outside the cell near the normal set point. To study this question, living neurons from rats were genetically manipulated to make proteins that generate light as calcium levels inside the cell change, or as neurotransmitters, the chemical signals are released. The function of individual synapses was recorded with microscopes. I found that slightly decreasing calcium levels outside the cell below the normal value can cause many synapses of a neuron to completely shut down. This all or none behavior caused by calcium levels outside the cell is unexpected because it shows that calcium coming into a neuron is controlled by a sensor that selectively turns off synapses. This discovery shows that the normal set point of calcium levels outside the cell has a critical role in controlling how neurons communicate because synapses are susceptible to shutting down from any other change that would lower calcium entry. Moreover, these results will impact future research to study how neurons communicate because it highlights that the choice of calcium level in experiments is very important to how synapses behave.